Welcome to our lecture series 8. We were discussing about ultraviolet spectroscopy instrumentation. The instrumentation part 3. Through this lecture series spectroscopy, we discussed what is an electromagnetic radiation, what are the properties of light, what happens when the light interacts with the matter, and Beer-Lambert's law, which is the basic principle of ultraviolet spectroscopy, and the excitation energy levels. And we have started with the instrumentation. We discussed light source and gratings. Now we have to recap and understand the monochromators. So when we talked about light source, tungsten film lamps and the hydrogen or deuterium lamps are the most widely used and a suitable light source as they cover the whole ultraviolet region. In some way, the halogen lamps also used above 375 nanometers, but hydrogen deuterium have a wide source. Tungsten filament lamps are rich in red radiations. More specifically, they emit the radiation at 375 nanometers, while the intensity of hydrogen deuterium lamps falls below 375. For above 375, we usually go for the halogen lamps. While we're talking about monochromators are generally a prism type or slit types. Most of the spectrophotometers are double beam spectrophotometers. So the radiation emitted from the primary source is dispersed with the help of a rotating prism. The rotating prism, we have a continuous rotation, and one region it be opaque and another region it would be transparent. And the continuous rotation and splitting takes place. The various wavelengths of the light source, which are separated by the prism, are then selected by the sills. Such the rotation of the prism results in a series of continuously increasing wavelength to pass through the slits for the recording purpose. The beam selected by the silt is monochromatic and further divided into two beams with the help of another prism. One beam will pass through the reference, another beam will pass through the sample. So both the beams get analyzed by the detector, will give the final result of our required things. So this region, that is monochromator, which includes the entrance slit, the dispersive medium, either it be a grating or be a prism, and the exit slit plays a very important role in selecting the wavelength, and light source also plays a major role, which helps to give a region, which gives a region in which the light can be produced to a desired wavelength. So in the next part, we will be discussing about the sample container, the detectors, and applications. Thank you.